Welcome to this lecture on the diversity of chordates. Today, we are going to explore a fascinating microscopic creature known as the Tornaria larva. Think of a beautiful butterfly and the crawling caterpillar it once was. In the vast ocean, many marine animals have a similar baby phase. We will uncover how this tiny, transparent, water balloon-like organism holds the secret to our own evolutionary history. Now that we know what our topic is, let us dive into what exactly a Tornaria larva is. It is the free-swimming planktonic larval stage of hemichordates, most notably seen in the genus Balanoglossus. Imagine a tiny, transparent oval spinning in the ocean currents. It is equipped with complex, looping bands of tiny hair-like structures called cilia. These cilia are not just for show. They are essential for swimming and filtering microscopic food. Despite its microscopic size, it possesses a fully functional digestive tract, including a mouth, stomach, intestine, and anus, allowing it to survive long journeys before metamorphosizing into a bottom-dwelling adult acorn worm. Understanding its anatomy leads us to a massive discovery in evolutionary biology. Why do we care so much about this microscopic ocean wanderer? When scientists first discovered the Tornaria larva, they actually thought it was the larva of a starfish. It looks almost identical to the Bipinnaria larva of echinoderms. This striking anatomical similarity is the primary reason scientists realized that chordates, the group that includes humans, are more closely related to starfish than to insects or earthworms. Without studying the Tornaria larva, the evolutionary bridge connecting simple invertebrates to complex vertebrates would remain a confusing mystery. To survive its long journey, this larva must feed constantly, which brings us to the mathematics of its survival. Biologists use the clearance rate equation to calculate how much water the larva filters. The formula is C equals A times V times E. Here, C is the clearance rate, or the volume of water filtered per unit of time. A stands for the surface area of the ciliated feeding band. The variable V represents the velocity of the water current generated by the beating cilia, and E is the capture efficiency. If the cilia beat faster, velocity increases, and the clearance rate goes up, meaning the larva gathers more food. Let us see this formula in action with a concrete example. We are given a Tornaria larva with a ciliated band area of 0.05 square millimeters. The cilia generate a water velocity of 2 millimeters per second, and the capture efficiency is 80%, or 0.8. We want to find the clearance rate. We substitute these given values directly into our equation. Since all units are in millimeters and seconds, we multiply 0.05 by 2 and then by 0.8. This gives us a final clearance rate of 0.08 cubic millimeters per second. This is the tiny droplet of water completely stripped of food every single second. Now that we have the clearance rate per second, we need to scale this up to understand the larva's daily life. We use the total volume formula, V equals C times T. In this equation, V represents the total volume of water filtered over the entire time period. The symbol C is our clearance rate that we just learned how to calculate, and T is the total time in seconds. This simple multiplication allows us to scale up a microscopic per second rate to a macroscopic hourly rate. If the time increases, the total volume filtered increases proportionally, which is a fundamental calculation for understanding larval survival and energy intake. Building on our previous calculation, let us find out how much water is cleared in a full hour. We know our clearance rate is 0.08 cubic millimeters per second. We are asked to find the total volume over 1 hour, which is exactly 3,600 seconds. We multiply our rate, 0.08, by the time, 3,600. The result is 288 cubic millimeters per hour. This means that despite its microscopic size, the Tornaria larva is an incredibly efficient feeding machine, processing hundreds of times its own body volume in water every hour just to survive and grow. Feeding is only half the battle. The larva also needs to travel, which introduces us to larval dispersal. The Tornaria larva uses its teletroch, a thick band of cilia at the bottom of its body, to swim upward toward the ocean surface. However, the ocean is not still. Strong ocean currents constantly push the larva horizontally across the sea. Because the larva is swimming vertically while being pushed horizontally, its actual path through the water is a combination of both movements. This delicate balance between biological effort and physical ocean forces dictates how these planktonic larvae disperse to new habitats across the globe. To understand this combined movement mathematically, we look at the resultant velocity formula. The formula is v sub r equals the square root of v sub x squared plus v sub y squared. Here, v sub r represents the resultant velocity, which is the actual speed and direction the larva travels. The symbol v sub x represents the horizontal push of the ocean current, while v sub y represents the vertical upward swimming effort of the larva. If the ocean current becomes stronger, the horizontal velocity increases, which in turn increases the overall resultant velocity, pulling the larva faster across the ocean. Let us apply this formula to see exactly how fast the larva is moving. We are given a vertical swimming velocity of 1.5 millimeters per second and a horizontal ocean current of 4.0 millimeters per second. We need to find the resultant velocity. We square the horizontal velocity to get 16.0 and square the vertical velocity to get 2.25. Adding these together under the square root gives us 18.25. Taking the square root yields approximately 4.27 millimeters per second. This shows that the overall journey is heavily dominated by the horizontal ocean currents. Now that we have used this formula, let us understand where it comes from conceptually. The vertical swimming and horizontal current are completely perpendicular to each other. Because of this, they form a perfect right angle. We can treat these velocities as vectors, which have both magnitude and direction. By placing the horizontal and vertical vectors head to tail, they form the legs of a right-angled triangle. The actual path the larva takes is the hypotenuse of this triangle. According to the Pythagorean theorem, the square of the hypotenuse equals the sum of the squares of the other two sides. Taking the square root of both sides gives us our resultant velocity formula. Having understood the math behind its survival, let us look closely at the anatomy of the Tornaria larva. On the screen, you can see a detailed diagram of this transparent, oval-shaped creature. At the very top is the apical plate and apical tuft used for sensing the environment. Notice the complex, looping, circumoral band of cilia that we discussed earlier, which sweeps food into the mouth. The food then travels through the esophagus, into the stomach, and out through the intestine and anus. At the bottom, you can clearly see the teletroch, the thick ciliated band responsible for the vertical swimming effort we just calculated. We have covered a lot of ground today, So let us summarize the most important ideas. First, the Tornaria larva is the crucial planktonic dispersal stage for hemichordates, allowing them to populate new habitats. Second, it relies on complex ciliated bands to efficiently filter food and navigate ocean currents, which we can model using clearance rate and vector mathematics. Finally, and most importantly, its striking anatomical resemblance to echinoderm larvae provides undeniable proof of the evolutionary link between starfish and chordates. You now have a solid understanding of both the biology and the biophysics of the Tornaria larva. Great job!